So, welcome back, everyone. Thank you so much for taking the time to join us at Global Genes Live, Part 2 of Rare Patient Advocacy Summit. We are excited for this session entitled Are Oligonucleotide Therapeutics Right for Your Disease? So, my name is Rodney Samago. I'm an assistant professor at the Department of Molecular and Human Genetics at Baylor College of Medicine and the Jan and Dan Duncan Neurological Research Institute, and I will be your moderator for today's session. So before we get everything kicked off, I want to review a few brief housekeeping notes. First, attendees are currently muted by the moderator to reduce any interruptions. We encourage you to use the chat function in the On24 window for any questions you may have throughout the session, and the moderator team will present them to the speakers later. So to open the chat, please click on the blue chat button located on the meeting controls at the bottom of your screen. If there are questions that are not able to be addressed by the speakers or we run short on time, which hopefully we will not today, we will do our best to follow up after the program. So also make sure to post any of your top takeaways from this session and fun moments if you're taking pictures at home on the Global Genes Live social media channel and tag @GlobalGenes for a chance to be featured on our social media channels. And the official event hashtag is #2020GG Summit. Finally, we encourage you all to participate as active listeners and take advantage of an opportunity to hear from these leaders here and experts to ask questions of them. This program is now your time to learn and find answers on critical challenges in the rare disease space. So with that said, I'm now pleased to welcome our discussion leaders. So first, Daniel O'Reilly, postdoctoral associate at the Kovrova Lab at the RNA and Therapeutics Institute at University of Massachusetts Medical School. Then we have Christina Bauer, Vice President of Patient-Centered Drug Development at Ionis. And finally, Sanat Kumar Ramesh, founder of Cure GPX4. They will each share a more complete background as we delve into the conversation. But for now, I'd like to invite Daniel to kick things off.
Thank you very much for the kind introduction. So first, I'd just like to explain a bit more about who I am. I am actually, let's go back. I am, does it keep changing? There we go. So now everyone can see the slide. So I am actually a patient advocate first before I am a researcher. My mum unfortunately had Huntington's disease as I was growing up, and this caused me to undergo progress differently in two different ways. The first was to start a charity with my family to raise money for patients of Huntington's disease and their carers. And the second was to go on the basic research side and look into oligos as therapeutics for rare diseases. And I did a PhD at McGill University to look at oligochemistry. And then now I'm working as a postdoctoral associate at the RTI, really trying to translate basic research to clinical applications. So, but what you're really all here for is what is an oligonucleotide? And at its heart, it's just an oligonucleotide is a string of the bases A, G, C, T. And these are the bases that we all know make up DNA. DNA is an oligo. It's just many of these bases joined together into a sequence. And what's great is these sequences can be tuned and changed to target different pieces of RNA in your cell. And there are many different methods of doing this, one of which, the most famous of which, is the antisense, is the single-stranded piece of DNA which targets the RNA of interest, and then it can cleave the RNA or modulate splicing that way. The second of which, and is more up and coming, as the small interfering RNA, which actually uses double-stranded RNA and then loads into this complex called RISC, and then acts a similar way to the antisense on the target RNA to silence it completely. The final way is actually gene editing, which you've probably all heard of, the CRISPR. This CRISPR system actually uses an RNA oligonucleotide to find the target DNA of interest. So they're really, really, really important. The problem with this is that if I was just to make a piece of DNA or RNA and inject it into our bodies, it actually wouldn't work. Your immune system has millions of years of evolution designed to protect you from foreign pieces of DNA and RNA, which is a great thing, but it's not great for a drug. It won't get, the RNA will not get taken up by cells, and it will also be easily degraded. So what I do as a chemist is we make changes to the DNA and RNA so that it's no longer recognized by the immune system. It's no longer recognized, much more stable, has better uptake in cells. And actually, chemical modifications to DNA are in all therapeutics, whether it's ASO or siRNA approved therapeutics. Chemical modifications are in all of those. I'm also I'm a trainee rep on the board of directors of the Oligonucleotide Therapeutic Society. On the left, you can see our board of directors. They're all great experts in the field and really a bunch of kind people. On the right is the events innovation team we have. These guys do all the logistical work and all the planning for our yearly conference and as the society as a whole. Without them, we wouldn't run as well as we do. Recently, this society is really big for therapeutics, and recently we've had a big push to have more patient advocates come to our yearly conference. And last year we had Elizabeth Room, and actually this year's conference, that was a week ago, we had a whole session on patient advocates and N-of-1 type diseases. We have a couple of resources that you might want to be aware of. The first is a webinar that you can go and listen to about oligotherapeutics that's designed for non-scientists. And the second is a briefing document that goes through the step-by-step of what it really takes to make an oligo therapeutic for a rare disease. And so thank you. Yeah.
So with that, we'll move on to Christina. Christina, are you ready to?
I am. Yes. Can you hear me now? Sorry about that. I'm just trying to make sure because I can't see the slides per se, but I just want to make sure that you can. So thank you. Are we good?
Yeah, I think so. Let's see.
There's only a couple. It should be slide 13. How about if I just announce which slide it is before?
Sure, I got it. Yep, here it is.
Okay. Apologies. So thank you for having us. Just to give you a brief intro to myself. So patient-centered drug development at Ionis is really focused on looking for mechanisms to incorporate the patient voice throughout research, drug development, and all the way through access for patients. And so that's really the root of what my focus is, focusing on the patient. And I know this session's about our antisense drugs, the right approach for you. We also do help all of the incoming inquiries that come in through Ionis to facilitate those research questions as well. So really what we're here to talk about is how do we translate exciting scientific discoveries to help create therapies that will save patient lives, really, and antisense is just one approach to that. Slide 14. When we think about from the gene to, you can see it, okay. From the gene to discovering of therapeutics, there's a lot of work that needs to be done to characterize a gene in order to better develop a therapy and turn that target around that target, SMA is a great example of what can be done with antisense. And while I show this slide because it took 20 years, I think what's important to understand now is that we're living in a day and age where there is continuous technological advances happening, and we have whole genome sequencing that can help with diagnosis. And so the timelines that we're looking at are starting to shorten. And antisense is just really one way to take advantage of those advances. Slide 16. So antisense can uniquely address challenging diseases because they're broadly distributed throughout the body, right? We know that antisense drugs go to the liver, kidney, muscle, spleen, and for neurological disorders in particular, there's a distribution to the spinal cord and to deep brain tissue. We also know that antisense drugs are specifically, have precision and specificity, and so they can target a protein isoform or a gene variant. And antisense are creating an opportunity to target previously undruggable targets. So a great example would be toxic RNAs. And an example of a disease would be the role of DMPK that accumulates in the nucleus for myotonic dystrophy, and that is one target that we are looking at. And as you heard from Dan, right, antisense works through multiple mechanisms to either increase or decrease protein production, and it can also modulate splicing to produce a missing protein, which is what the case was in SMA. Sorry, I'm having a hard time not seeing the slide. So if you think about, and you can move to slide 17, the genetic basis of disease, you know, what is it that we need to understand about your disease to understand if antisense is the right approach for you? And the genetic basis of the disease is really a key factor, right? Understanding what the root cause of the disease is, the mutation or target that's involved, and specifically so we can translate that genomic information into a drug. I think that is the basic step. But then there are other components that we need to understand, like where in the body to target the therapy, how or where does the disease originate, how does the disease progress, and what organs are actually affected. So characterization of disease of the disease-causing gene is also very important. Understanding the role of the gene, and I was trying to think of like different examples. So for MECP2 duplication, right, there is too much MECP2, so you need to lower it. But yet, if you think about that target, MECP2, not enough of it is Rett syndrome, causes Rett syndrome. So there's many examples of why it's so important to understand all of these little details about your disease. And then really understanding the primary mechanism of disease is another critical component, because we need to understand if there's a gain of function or a loss of function. Do we want the gene to go up or down? And we need to know what direction that is. And sometimes I think that's really the hardest information to understand with novel mutations and targets. But that actually makes a huge difference in the approach that we take. And we can't really get started until you know what you're trying to tackle with antisense, because they're very different drug discovery paths. And so I just wanted to use this intro as an opportunity to kind of throw a lot of ideas out there and concepts of what are the things that we're kind of talking about today that we need to better understand, I think. And with that, I will stop and turn it over to Sanath.
Thank you. Cool. So I'm Sanath Kumar Ramesh. I'm a father of a 2-year-old who has an ultra-rare genetic condition due to a mutation in a gene called GPX4. And outside of my personal life, I work at Amazon as a software manager in the healthcare division. And quite honestly, my life wasn't this way before. It was primarily, traveling the world, going to different places, and until my son was born with this complication. And in the first few hours of birth, we knew there was something wrong with him. Even the nurses could tell that something was wrong with him. And we spent the next 21 days trying to find a diagnosis for his condition. We did a whole bunch of tests, and sort of everything came out negative and inconclusive. And we came back home, and he continued developing. He continued to smile all the time. He's a happy little charming kid until his first birthday. That's when we got the news that he has a mutation in a gene called GPX4, which is causing all of his skeletal abnormalities, his neurological abnormalities. He still doesn't have the head control to be able to sit, or the muscle control to be able to stand or walk or even hold a toy and drink anything or eat anything orally. And so, since then, after we got a diagnosis, it was sort of a turning point for our lives, because we now had a target that we could at least pursue at. And so we wrote a blog post explaining our story, which sort of went viral, and we raised about $100,000 in 2 days. And that was sort of the turning point, again, another turning point in our journey, because there are thousands of people around the world who sort of believed in our ability, to deliver, and also wanted us to succeed. And so at this point, we really had to come up with a strategy for our foundation, for our therapy development. And my approach since the beginning has been, what is the fastest thing that we could do to get something for my son? And the fastest that I could do is to walk down to the CVS next door and get a drug for him. And something short of that is trying to do exactly that tomorrow. And so now I need to really find which of the drugs, thousands of drugs that CVS has next door that will work for my son. And so I did some studies in the literature and looking into several drugs that could be repurposed for this condition. And we identified a couple, and we started my son on, we identified 36 drugs, and we started my son on 4 of them in last September. And after we started the first drug, we saw a significant improvement in his ability to lift his head up from a tummy time, which he has never done in the last year. And after about 9 months of him being on this cocktail of 4 drugs, we saw a reversal of some of his skeletal abnormalities. And these skeletal abnormalities were actually the hallmarks of this disease that led to the diagnosis. So we were even our orthopedics doctor is kind of was astounded by seeing this result. The key here is that we don't really know which of the 4 drugs are working. We don't really know. We didn't have a scientific process of coming up with the concentrations or the dosages for each of these drugs. But we said it's better than giving anything to mice. Let's give it to my son first. And I think the strategy that I'd been following all along is trying and going closer to human first before I give anything to a mice. And that strategy sort of led me to first, identify drugs that we could repurpose. We also got my son started on an experimental medication in August this year. And I'm also currently looking at gene therapy and ASOs as an option for my son's condition. And along the journey, we have already built the animal models necessary to be to keep the option open for both gene therapy and ASO. We had made significant investments in building the right models and creating fibroblasts and all of those things necessary for us to be able to keep the option open for any sort of therapeutic approach coming down the line. It could be gene therapy, it could be ASO, it could be CRISPR. But my strategy has always been, what can we do today to get us the treatment fastest? And that's repurposing, followed by investing in the models necessary to be able to open us up for all therapeutic options.
All right. Thank you. Those were all wonderful talks. So I think what we'll do now is proceed with the Q&A session. And I think the way we can do this first is based on some of the, let's say, topics and themes that we have covered in our own discussions, but working backwards. And each one of you could address these, I would say, thematic areas and go from there. And then we'll also take questions from the audience. So what would be some of those deciding factors that you can share amongst the 3 of you with the audience with respect to you trying to understand disease mechanisms for their specific disease areas? Do you have any key pieces of advice?
I can start probably giving you the patient foundation point of view, which is start from the literature. And I think this is interesting because there's probably a lot already known either about the gene or about the pathway. And if it's not known about the pathway, look at the subcellular parts that this protein is sort of impacting on. And that essentially gives us a reasonable understanding of the disease mechanism. It doesn't give us precise understanding to be able to go to the FDA and say, here's how it works, but it gives us a reasonable understanding to unblock and decide on which is the next steps you could take in terms of building a therapy.
Dan or Christina want to address?
Sure. I was just going to say, we did talk about the idea of having like research champions, right? Like Dan has, a driving force behind what pushes him to research for Huntington's and the antisense work that he's doing. And if you, I think that's a place where people could actually reach out and find their own research champions to help them in their efforts to really get a better understanding. And those relationships, I think we should think of researchers as much more approachable than we might want to. So as you're looking through publications and as you're seeing the names of the people that are researching the disease and the genes and the targets of your interest, I would hesitate to reach out to them.
Yeah, I totally agree. Like, you might find if you're looking at a particular gene, let's say, at first, if that gene's not been studied for a disease, there might be no therapies for it. So at first, you might find, let's say, a biologist who's been working pretty hard on the gene. So go to them, and then they might have some answers for you about the gene and how it works in the cell and the body. But then you can also reach out to Oligotherapeutics people and be like, okay, we have this researcher, and then you can start to almost build a team, a network of people. Because it necessarily won't always be one researcher. It'll be like a network of people, including industry people as well. Like, that's the other thing. Industry.
I like that actually. I like the network aspect actually, because everybody's, researchers have their own focus, and they're super deep into and specialize in their focus. And it probably will require multiple people to look at it from multiple angles to then start talking about what are the actual approaches that we can take forward. So having more than one researcher that is covering the same problem from multiple angles might be actually a very viable strategy.
Yeah, sure. I mean, take myself. I'm such a chemist that a lot of the underlying biology, it'd be better if you come to me and talk about therapy for us to go and talk to an actual biologist. Yes, I do some work on biology, but, I don't have the same experience as someone who's done a PhD plus postdoc with bio stuff, for sure.
Right. And we're all open to collaboration. I think that's the key, right? Is that we are all open to those collaborative efforts. And that is something that's kind of the basis of what's, driving the discoveries forward, I think.
And I think you bring up the next point, which is a very, I would say, a timely aspect related to our theme. So when we think about the biology and think about networks of collaborators, whether they span stakeholders within academia or industry, pharma, et cetera, how would you advise to better de-risk and delineate what those risks might be when trying to impart advice or knowledge to patient advocacy leaders and groups, thinking about oligonucleotide therapy, whether it is right for the condition as opposed to whether it is right for my person, my people, my child, et cetera.
Mm-hmm.
Are you asking me?
Actually, I can take it. I'll take the child part, and someone else can take the science part. So, my rationale. So everybody has a personal perspective on what you want to do for your child, and it's actually very different from different people. So even within a couple of families that have the child with the same disease, they'll have a different comfort level on whether they want to try an experimental drug or not. For example, I was chatting with another parent who was on an SMA trial, and they didn't know if their child was getting a placebo or they were actually getting the drug. but it's a chance that they want to take, they're willing to take, and it's an experimental drug at that point, and there's a chance that something might go on. And so to me, it's been a huge battle that we are facing as a family to determine what level of experimental therapies are we comfortable giving our child to, and as much as we are pushing hard on research, we are also very conservative when it comes to taking on an experimental therapy that doesn't have strong safety profiles. Now the question is, how do you really understand what the safety profiles is? And this is where the network of researchers that you build trust with, and that you can trust them, are there to advise you, and the doctors are there to advise you. So even though on the one hand, professionally, you're sort of pushing research hard to get a treatment for this condition, it doesn't mean that you really have to give it to your kid unless you and your family are comfortable. And it's a family decision. It's a different decision than a scientific decision.
Right. I mean, the scientific decision, right? You want it to have a solid basis, right? You don't want to just look through the literature, find an RNA and just pick, 5 sequences to find that antisense drug, right? I mean, that's the other concept that we really need to talk about. It's not, we screen thousands of compounds for each RNA, and they're not all created equal. And so I think that's another important thing to really think about in the approach that you're taking.
And Dan, do you want to add anything to that?
Yeah, for sure. I think when you have conversations between the families and the researchers, it's about being open to the challenges and what will need to happen. For example, with the clinical trials. In the clinical trials, unfortunately, for the trial to work scientifically, some people will have to take the placebo, will be willing to have to take the placebo in comparison to the others that will get the drug, but it's needed so that everyone can gain from that. And then with the research, the basic research, like Christina said, you can screen thousands of compounds and not get hit. And it might not happen. And I think just being open about that, like as we are, that's how my approach is trying to just be open and be like, look, we can try it. I'm always down for trying stuff, but it might not happen. You might not find a sequence for antisense that works for you.
Right, and I think that also raises an important, I would say, consideration when thinking about these different modalities with respect to time. So you basically underscored one of the main issues, I would say a very pressing issue of urgency, and time is the number one, non-renewable resource. One can always earn more money, one can always try to get more things, material things, but the time cannot be replaced. So how do when you interact with individuals, either within your foundation or your company or your institute and others, how do you advise them? What I would say nuggets of key advice can you give when it comes to balancing that time issue?
I think from my perspective, just there are things that can happen in parallel to the research, right? So while that drug discovery is actually happening, there are components of understanding the disease and different aspects of the disease so that can be translated into a more efficient design, into better selected or fit for purpose kind of endpoints that are better suited for a specific rare disease. So there's definitely a lot of work that can be done in parallel, and that saves a significant amount of time, I think, going forward, because that changes the length of a study that it would need to run. It removes a step that we would as industry might do after. Right now, we're all thinking about collecting natural history data and longitudinal data because that's critical into our development program. But it's, for some, ultra rare diseases, sometimes there aren't that many patients. And so if you're forming a patient group and empowering yourselves to collect your own data, I think that's critical information that really accelerates that timeline.
I like the parallelism aspect there, because the key to there are a lot of activities that we have to do to get to a therapy. And some of the activities are long poles, like just developing a mouse model is going to take you 14 months, 16 months, right? Just get them started initially, even if you know, or if you don't know if you need them. And similar to the basic science investments, like as you're pushing the therapy development forward, invest in basic science. And it's one of the lessons that I've learned is I didn't invest enough in basic science. Now I've lost time that I could have actually learned more about the biology before I really have to really need it. And so find the long pole activities and get them started earlier.
Yeah, I totally agree. Something else that can be done at the same time in parallel is actually funding. If the researcher is doing his job or the industry people are looking for a sequence design in the mouse models, like you could be actively looking for funding opportunities and sending that, rather than developing something and then having to wait months and months to find out if you get a funding opportunity to then take it forward. It'd be better to do that in parallel as well.
Okay, great. So I've been taking notes here. Oh, did you want to add something, Chris?
I started to read the questions.
Oh, yeah, I was going to go to the questions too. So let's take the audience questions. And so we'll start with the first one. Okay. So let's see. Question number 1. How are researchers approaching therapies with the potential for patients with multiple rare diseases? Are there any examples of that? Who wants to take
I've
this?
only come across a couple, and I was quite, it was a homozygous familial FH patient that also had, I believe, myotonic dystrophy. So I'm sure there are more examples out there, but I wasn't expecting it, honestly, but I'm sure there are plenty of examples. But I think each disease would have to be targeted independently because, right, they're very specific stems of origin for each of those.
I mean, maybe it could be reframed in the sense that the clinical diagnosis, so say, for example, Cornelia de Lange, so there are many genetic variants that could contribute to that. So perhaps are there things that other examples that you could think of, Dan or Sana?
Again, I think it comes down to that network of people. If it's multiple mutations for the same gene, like Duchenne muscular dystrophy, that's another one that has multiple mutations. And, I see a lot of people, it has a lot of different mutations across different exons. And so I see some people working on exon 53, for example, some people working on exon 51. And it's really that, like, allowing people to take on each individual gene and then having them talk to each other, I think is the key to that.
And if the diseases are due to like large chromosomal deletions, then that classifies into another bigger classification of diseases. And there are some chromosomal deletions that sort of connect to other chromosomal deletions in terms of phenotypes. And at that point, it's not a specific gene that you're looking at; it's specific phenotypes that you're starting to match and connect the diseases together. But that's the example that I've seen. But I don't really have. I've not seen any specific therapies that have been developed targeting both.
I have 2 questions for you, Sana, from the audience. So how did you find a doctor who was willing to work with you on experimental therapy? So most doctors may be hesitant or somewhat risk adverse, right, when it comes to that.
Yeah, and I think the key is to find an MD PhD, and I've had other patient foundation leaders also tell me that as well. Is when you have someone who's an MD PhD, they're also looking at the research angle as well, and they understand that it's not clinically treatable, but from a research setting, there's good alignment with the science on why this drug is better for this disease. So it takes. I'm not going to say it's easy. It takes a lot of convincing, and I've tried to find multiple doctors that would work with me on that, and it's a challenge. But the folks that I've found to be best are the MD PhDs that sort of understand the research aspect as well.
So then that kind of is a callback to the champion, kind of someone who would advocate. But this is related to another question. So would you say that was the primary driver of how you arrived at the drugs that you gave your son.
It was in collaboration with them. So we did our own research. I did some research. My doctor did some research, and others in the research network did some research as well. But it was, at the end of the day, a collaboration between us and the physicians, and the level of comfort they had with administering the drug. And it's when the physicians are part of a larger system, like a hospital system, then that also, their opinions also come into play. And the hospitals have usually an IRB that have a say on what drugs can be given or not, if it's an experimental drug, or if it's not a supplement. For example, supplements and other things, the doctors have a lot of leeway to be able to give it to your kids. And so it took us about, we got, for the experimental drug that we tried, we got the paperwork ready last November, and it took us until this August to actually get it through the FDA, through the IRB, through the hospitals, and COVID happened. And so it's at least 9 to 12 months of effort, of paperwork effort.
Christina, did you want to add to that? I thought I heard you.
I was just going to say, the other thing that made me think of, right, is really target physicians that are specialists in diseases where there's a lot of symptom overlap, right? So there's a level of familiarity that they may be able to translate into your disease as well.
Okay. That's all very good advice. Okay, moving on to the next question. Is there a potential for a single gene mutation in an autosomal dominant disease in skin? Are there topical therapies for this? Maybe Christina? Or...
So we don't necessarily have topical programs right currently. We have in the past done topical administration. So it is something that is possible.
Yeah, there's none approved currently that are topical, but
no,
I don't think.
Not that I know of. I agree.
So this is an interesting question, the next one. So 10 years isn't feasible for most people, and I think we all would agree with that. Is there any chance of accelerating these therapies? What are your current thoughts related to that?
I think there are. I'm very optimistic. Go ahead.
Yeah. I totally agree. I mean, if you look at Tim Yu with Millicent, in that case, he found a drug. He had it injected into Mila in a year, was it? I think, something like that. So it can be done, but it is very dependent on your disease and what you have. Like, for example, in that case, I think they very quickly got or had the mice models and the cells to do a bunch of the data and test. And it was going to be just for her, not more people, not more than one person as well. And so the FDA were okay with it. But if you have got a disease that you only know the sequence mutation and you don't know anything else, then it might be more difficult to translate that in such a short space of time. It might take longer. But yeah, for 10 years is a lot.
Right.
Yeah,
and I think that
Okay. So realistically speaking, you want to separate out what your expectations are when you say, can we accelerate this? Is this for one patient? Is this for 10 or say 30 patients? Or is this for a disease that have thousands of patients, right? And depending on that, the complexity changes. For one patient, there's obviously ways to accelerate that. There's the FDA has the single patient IND, or what they call a compassionate use program, which allows you to sort of dose an experimental drug onto a patient, given that there's no other approved treatment, as long as you can show the science behind it. And that's what I think Tim Yu would have actually done that as well with Millicent, and that's what we did with our kid as well. But the challenge is, even if you were to develop a therapy at a very, very fast pace, let's say one year or 2 years, if you had more patients, then going through a clinical trial is, again, a longer process. And that's where things get a little bit more challenging. Patient foundations typically have the money to be able to develop the therapies, or they can raise the money to develop a therapy. But once you get to clinical trials, you probably need like several millions of dollars, I don't know, 10, 20, 30 millions of dollars or whatever that needed. And at that point, finding an industry partner who can work with you to get that going would be the best bet. And that's where I think the long pole ends up being.
Good. So we have 15 minutes left, and I want to get through some of these questions that remain. So this is a two-part question. Do ASOs work better for gain of function versus loss of function? And this is specifically thinking of CAC and A1A variants. And the second part of that question, can you clarify gain of function versus loss of function? And researchers sometimes use these in different ways. Who would like to tackle that one?
I can start, and then Dan can probably correct me if I do something, say something incorrect. I mean, gain of function is just means that you're overproducing a protein, right? And so you have too much of it, so it's doing too much of whatever it needs to do. And loss of function is the exact opposite. You're just missing something that's needed, and without it, that disease process gets initiated. So, does it work better for one or the other? Not necessarily. It works great for both, right? It just depends on the target and that role of that target within the disease state itself, right? What is that target? What function is it controlling? What other targets is it affecting? And the basic biology of that is where that comes in and plays a role. But it works great both ways.
Yeah, for sure. I totally agree with that. I also have heard people say like gain of toxic function, like with Huntington's, where you don't have more of the Huntington protein, but it gains a toxic function. And so you can have that situation as well.
Right.
Again, totally agree with Christina about that. You don't necessarily know whether the ASO will work better for one or the other. There's no predictor for that. You just have to try it.
Mm-hmm. Mm-hmm.
But is there, when there's a loss of function, do you still expect ASOs to work? Because ASOs depending on how what the target is and what the role of it is. So for SMA, right, it was a splicing mechanism. You could correct that, and then you got a full transcript so that you could make a protein that was missing. So
yeah,
it just really depends.
Yeah. There was that second gene, right, that also produced a similar but slightly different non-functional protein, and then by skipping it, you produced a functional protein. That might not exist for other diseases, you know, and skipping might not work to produce a functional protein.
Right, so but there may be.
Go ahead.
I was just going to say, well, and he's correct, right? So there may be downstream effects or upstream effects that are really the main target, that rather than the gene itself that you're thinking that you're initially going after.
Mm-hmm.
Okay, so this is at the level of kind of collaborations, kind of the operational considerations and logistics. So how do you suggest to unite researchers of different fields around a huge gene that has so many different disorders attached to it? Any suggestions?
This is going to be an interesting one. So I'd say, I mean, everybody, if it's a huge gene, if it has a lot of disorders associated with it, I would expect that there's already a lot of researchers working on it. So you don't typically have the problem that other patient foundations have, which is there's nobody working on it. You probably have the opposite problem, that there's too many people working on it. And I don't know if you need to really unite them, because if your goal at the end of the day is to get a therapy for a particular disease, then you pick the group of people that are really interested in that disease. Maybe because they have a certain interest in a mechanism that's causing the disease, or maybe because they really care about the disease and has a personal interest. But I think if you find a common thread that can unite a group of folks to align with what your goal is, I think that at the end of the day is what you should be looking at. But if your common goal is to unite the researchers together and have them start thinking about all the diseases as a whole, it's sort of a different problem. It's like creating a consortium problem, right? And that's a whole different segment altogether.
It's one of my favorite segments, though, right? It's a great opportunity through advocacy to unite industry partners that may actually be competitive partners, right? But could join in an advocacy consortium to further advance the needs for everybody. And I think that is a very important aspect that needs to be pursued.
Yeah, for sure. I also like the idea of, if you have so many people that work on a disease, and you could just start your own, like, do a yearly conference. You know, I say that because, like, we have weekly meetings, HD meetings in the department, and it brings all the people who work on HD together. And so you could start by doing that for the disease and gene, and that would get conversations going.
Okay, so I have another one here. If a patient advocacy group has a patient registry of patients with genetically confirmed patients, how would you suggest we move forward? How do we find someone to screen compounds for our particular mutations?
So if you're looking at screening compounds...
Say again?
Yeah,
I'll start with this. So if you're looking at screening compounds, there are high throughput screens that you could do, and once you have the registries, you probably don't really care about a lot of patients. But if you can get fibroblasts from a few of the patients, then you can set up high throughput screens at different institutions. I know NCATS has a high throughput screen facility. I know like CROs like Charles River also does high throughput screens. Several labs actually do their own high throughput screens as well, and you could start there as a mechanism to do a screen. An alternative would be to do a phenotypic screen where you start by creating different animal models of the mutations or of the genetic condition that you want, say a worm model, a fish model, or a fly model, and a mouse model, and then screen drugs on multiple animals to see which of the drugs are consistently being, therapeutic or beneficial on all of the animal models, and that just gives you a higher confidence to be able to screen them. So the screening aspect is sort of independent of the patient registry itself.
Did you want to add to that, Christina?
I was just trying to think of how, obviously you could always propose targets, right? It's again, going back to building that network of who's going to screen for you. And I would include everybody that you can think of, including multiple industry partners that may be open to such a collaboration.
Yeah.
So,
and to find people, you can always use resources like the OTS, the Oligonucleotide Therapeutic Society. That has a list of all its members. And if you email them and say, look, I have this disease, I have fibroblast, I just need someone to test it, it's easier for them to say, okay, let's send this around to our members. Maybe someone would help them out, And a lot of people are willing to help.
Right.
There's a very specific question related to a gene that's quite large. It's SBF1. And so it's too large to fit into AAV. So is an ASO available? The question, that's the first part of the question. And then the second part, this is a double autosomal recessive, one deletion and one duplication of each gene. One inherited from mom and one from dad on chromosome 22. Christina, are you aware of any ways in which one can target SBF1 or similar large genes that might be too big for AAV?
I don't, but that is like a question that I could submit. Like I would personally submit to research to get help on, right? So that is a resource, right? We could help. We could ask internally to see if there's somebody that has that experience. I don't know what the answer to that would be, though. I'm sorry.
I see a comment that just came in, actually, for Christina, building upon your comment related to building patient groups. What value could accrue to patients, researchers, manufacturers that are from a therapy-specific social platform or social platforms?
So what value comes from that? I guess I'm not completely clear on the question. But I think, pulling all of these different arms together, right? Like researchers, manufacturers, using different platform mechanisms, creating a louder voice and unity, I think, is something that does benefit patients and help create a louder voice. And I apologize if I'm not answering that correctly. But I think, from my perspective, it's very important to empower patient groups to really build their databases and collect their own longitudinal data and really better understand their disease and start sharing that information. And that's another mechanism to really kind of stimulate interest and building upon those networks again.
Dan, were you going to say something about that?
Yeah, I totally agree with Christina's comment. Like, I personally feel the power of patient advocacy groups from myself being a patient advocate, but also when I went and I was working on Friedreich's ataxia. I went to a patient advocacy summit and presented my data, and honestly, it was the best experience. And I got so many different perspectives on my research that I hadn't noticed because I've been so focused on like almost the little details, you know. But having such an overview of like, okay, how will this translate, is really important.
Mm-hmm. And then I have a final question here. Can ASOs be designed to only target the mutated gene, even if it is a single nucleotide change? Or would it possibly target both alleles? Are there a general consensus, or is there a general consensus on what happens in an allele? So that's a three-part question.
Yeah, so the first part is, can ASOs be targeted?
For a single nucleotide, yes.
Single nucleotide, I think it has been done. I don't know if it's been translated into the clinic doing that. It's very difficult to get that level of discrimination, but there's certainly work being done in that. I've seen people work on that for Huntington stuff. So it's certainly doable. Obviously, the discrimination you get between the alleles will be different. It's not guaranteed you'll get any discrimination, but it's doable.
Mm-hmm. And Christina, did you want to add to that part?
No, I think, that was part of the basis, as Dan mentioned, right? The Huntington program, it looked at multiple different approaches, whether to target allele specific or not. And so it can be done. What is the best approach depends on the experiment, right? And what happens when you actually, do the science to support that decision. So, but it is possible, I believe.
Yeah. And just to continue that, you might not need the allele selective. I think for the Ionis antisense for Huntington's, didn't they see that they actually didn't need to be so specific to the repeat, right? They could actually lower the like normal gene to such an extent that even then you still got like recovery of the phenotype. And so that you might find that out, and so you might need the selectivity.
Right, and even other diseases such as amyloidosis, right, where there's a gene and there are multiple different mutations and variants of that disease, right? Targeting just TTR alone is enough to address not only all of the mutations but even wild type TTR. So, I wouldn't say anything's a dead end until we know the biology, the biological basis for it.
Mm-hmm. Mm-hmm. So I've got a question here from William L. And I'm trying to find the question. My question on muscular dystrophy was missed. I don't know if you guys can see it, but let me know if you see it, or William L., please pass it along back into the group chat. We have a few more minutes, and I think that this has been a very informative and also very, I would say, just the beginning, introductory set of questions here that can be expanded. I would say even in classes, right? Some of these questions that we have are, when we think about how to work within the space, conceptual, but also technical, right? And also very pointed. But then there's also the aspect of, I would say, working together and collaborating and ensuring some kind of path forward towards actionable, go, no-go decisions for all levels, whether it be patient advocacy groups, families, family unit, or academics or industry partners. And so one question is for Christina, would you be open to being reached for additional questions?
Oh, sure, absolutely. You can email at patients@ionasph.com.
Okay. And how about the others, Dan, Sana?
Yeah, totally. Just email me at myumassmed.edu. email address. It's just daniel.o'reilly and then at umassmed.edu.
Yep, and my email address is sanat@gpx4.org. I'm always available.
Okay. So for anyone who has questions, please feel free to reach out to all the individuals here, including myself. My email is rodney.samaga@bcm.edu. Thank you very much for attending part 2 of Global Genes Live, and we look forward to hopefully seeing you again at another session in the future.
Awesome. Thank you.
Thank you.
Thank you.
Thank you.
Thank you.
Yeah.